Okay, so the next speaker is me. So I'm France Carrier, and I'll co-lead this session for the radiobiology part. I'm also a professor in radiation oncology, and I hope everybody can see my slides because when I'm sharing, I don't have a way of saying if people see it or not.
Yes, we can see.
Wonderful, thank you. Okay, so let me just go back. So the title of this lecture is Radiosensitizer, Radioprotectors, Bioreductive Drugs, and Chemoradiation Strategies. There's a lot of stuff to go over. We have objectives for each of those part of the lectures. I've divided that into three parts. The first part, I will cover radiosensitizer and bioreductive drugs. The objective, of course, we would like you to understand the role that each of those players have. In this, we'll talk about the chemical radiosensitizer, apoxic cytokines, and non-apoxic cell radiosensitizer as well. For the radioprotector, I don't know, can you see my pointer when I use my, no, I don't, okay.
Yes, we can.
Yes, we can.
Okay. Wonderful, good. So then the second part, I'll go over radioprotector, and then we'll cover some free radical scavenger. Mainly, a lot of emphasis is on the amifostin, so we'll talk about this one and a couple of others, including bacterial flagelling. And the last part, I will cover the radio strategy for chemo radiation and how we can combine the two chemotherapy and radiation. The overall goal, of course, we would like you to do well in this section, and then keep in mind that what I've mentioned before, for radiobiology, all those topics will be covered. What I'm talking about today, this is the combined modality called, including this, and can be up to 15% of the questions. I'm gonna today cover two of those topics, which is the chemotherapeutic agent and radiosensitizer, and the immune therapeutic immune therapy will be covered later this afternoon by Dr. Ferris. So when we talk about chemo radiation or chemotherapy, what we want to do is really look at the dose that we're using, either of chemotherapy or radiation, versus the incidence that we're provoking. And then basically what we want to look at is the ratio that we have between the effect that we have killing a tumor cells, but also minimizing the amount of damage it will have to normal tissue. And this is a sigmoidal curve that you can see that based on the probability of tumor control versus the normal tissue. So what we want to do is have as much distance between those two curves, and this is what gives you the therapeutic index. And so in that particular example, for example, for 50% tumor control, so you can get maybe 5 or 10% of normal tissue damage, and that would be considered acceptable depending of what the goal is to how much tumor control you think you can obtain. So how do we make advantage of or really distance those two curves? There's different ways we can do that. There's a time factor that, for example, we know that if you do hyperfractionation, so you're going to protect more of the normal tissue, so then and still keep the local tumor control. So you're going to increase the distance between those two curves. We can use also drugs that would increase the sensitivity of the tumor, that move this one to the left, or that can protect the normal tissue. And then we'll talk about radioprotector and then move this curve to the right. So essentially, this is a balance between radioresistance and radiosensitivity of tumor and normal tissue. And what are the factors that can affect that? If a tumor is very large, it's going to be more radio resistant. One of the reasons of that is because you get repopulation between the different fraction of radiation sometimes, and then you get accelerated tumor cells that would contribute to radio resistance. And I'll show you in a minute in a couple of slides that also that large tumor have a tendency to have poor vasculature, so that would lead to hypoxia, reduce the level of oxygen into the tumor. And then those tumors also have enhanced repair, and that would also lead to increased radioresistance. On the other hand, a smaller tumor would be easier to control with local radioactivity, local control. If the tumor is defective in one of the DNA repair pathways, so that would increase the damage and then make the cells or tumor more radiosensitive. And if the tumor has a good vasculature, again, that would increase the oxygen effect that we'll discuss in a minute. And then we can use some radiosensitizing agent to increase the sensitivity of the tumor. So the factors that really make a tumor more or less radioresistant or radiosensitive has a lot to do with the level of hypoxia that we have. And this is due because you have in highly proliferating tumor, the tumor are growing really, really fast, and they try to keep up with the demand of the tumor by generating a lot of blood vessels very rapidly. And that brings to a very chaotic situation where you have blood vessels that are not mature, that they have blunted end, they are leaky, and that leads to poor oxygenation of tumor. And those are like the pocket in light blue here, so that those areas will have a very little oxygen. On the other hand, a normal tissue where you have normal vasculature, and this is how the cells are distributed in a blood vessel, where you have the endothelial cells that are lining the inside of the blood vessel, and then we have the pericyte at the outside that are tightly joined together that make a very tight vasculature. And then when you have this highly proliferating blood vessel development, so then you have a lot of leaks there, and that makes very poor oxygenation for the tumor. So why is that important in a tumor and in reduced sensitivity? Because this is what we call the oxygenation effect, is that in the presence of oxygen, so then you can have the you can have the free radical that are formed by radiation that can be now coupled with the oxygen and then form an organic peroxide like the O2 that would permanently fix the damage of the DNA, and that leads to reduced sensitivity. If you don't have oxygen there, so that damaged DNA can be repaired, and then that would lead to radio resistance. So that's why it's important to have oxygen that is present, that to have good vasculature that can bring that and increase the radio sensitivity. And this is basically the level of hypoxic of solid tumor. This is the basis of why we're doing fractionation or fractionation radiation therapy. Because in any solid tumor, you're going to have a mixture of the cells that are oxygenated and cells that are hypoxic. This one's shown that it's in the middle, but it could be in different pockets of the tumor as well. So with each radiation dose, you're going to kill the sensitive cells, the one that are oxidized, and then you give some time for the next fraction for this balance to be reassortment of between the oxygenated and non-oxygenated cells. And then at the next fraction, then you have another proportion of cells that are oxygenated and will be sensitive. So this, and you keep doing that until then there's no cells. With each fraction, you're going to kill more and more cells that have had time to be oxygenated. So that's why the effect of hypoxia then is kind of minimal if you do several fractions. That would be a problem then more if we have, when we talk about hypofractionation, like with SBRT, then the effect of having the proportion of hypoxic cells in the tumor, then we'll have more consequence of the success of the radiation scheme. This is just, again, to put that into perspective, that between the tumor cells and then the vasculature that we have, how we can increase the oxygenation of the tumor cell by increasing the perfusion of the delivery rate, and if you have a better vasculature. So then you would think that if you use an anti-angiogenic factor, for example, that would prevent the development of blood vessel, that should not be effective as increasing the reduced sensitivity. But actually it is, because if you use an anti-angiogenic factor, you're going to prevent the formation of those rapidly developing immature tubules, and then you would reestablish part of the blood flow into the tumor that would increase then the reduced sensitivity. So that's why the addition of an anti-angiogenic factor could be beneficial to reduce or increase reduced sensitivity. And like I mentioned before, that would be more consequential for hypofractionated radiation. Okay, so why are the tumor cells surviving under those, if you have hypoxia, and how they really start making so much blood vessels and trying to compensate for this low level of oxygen? And this is all mediated by transcription factor that is a hypoxia inducible factor. You may have heard of that in different lecture, but it's good to mention that here. This is a very important transcription factor that under normal condition, it is rapidly degraded because it is the oxygen molecule then can activate the PDH or the prolyl hydroxylase, and then that would lead to the degradation to the proteasome of the HIF1 factor. Under hypoxia condition, then when there's no oxygen, the HIF1 can translocate into the nucleus and then partner with another protein, F1 beta, and then together they can recognize hypoxia responsive element on the promoter of several genes and then increase the transcription and translation of those protein. One of those is the VEGF, which is the endogenous growth factor for blood vessels. So now you can see that under hypoxia, this is the reason why you're going to see a high proliferation of blood vessels that are going really fast because of this factor here is upregulated and really lead the progression of those blood vessels. So that's the problem. So now what's the solution? Is there a solution? How do we deal with hypoxia to increase oxygen level into a tumor and increase radiosensitivity? One of the first things that people wanted to do, say, well, that's easy. We're just going to force oxygen into the tissue, and then we'll make it more radiosensitive. And then they start doing hyperbaric oxygen tube and putting people in there and trying to increase the flow of oxygen into, or force the flow of oxygen into a tumor with 100% oxygen. That was very cumbersome, very difficult in practice. Some people were claustrophobic. 100% oxygen is flammable. It was not an ideal solution. So what are the other ways that we can do? So we can use hypoxic cells radiosensitizer that would increase the radiosensitivity of the cells that are deficient or have low oxygen level. And then the goal of this is that we will be able to do that only in the tumor cells and not in normal cells, because normal cells don't have hypoxia. So one of the first molecule that was developed is a smaller molecule that can take the place of oxygen. They call hypoxic radiosensitizer, or they are oxygen substitute. And the criteria that we'll look for is that those small molecules will not be rapidly metabolized. They can penetrate further than oxygen to reach hypoxic cells. They are selective to sensitize the hypoxic cells. They are chemically stable. They are soluble in water and lipids, so that would allow them to diffuse even in the absence of having a good vasculature into the tumor. And they should be effective at conventional radiation dose. So one of those first molecules that was successful in cells is indolzimazole. So it has a neutral group on the position 2 here. Depending where you put the neutral group, then you give it a different name. So we have another one that I'll talk in a minute, that is 5-nitrozamide. So the way that those molecules then take the place of oxygen in a tissue is that they would accept the electron from the radical that is produced following a radiation therapy, and then transfer that to the neutral group, and that would produce a short-lived free radical that would be cytotoxic and then fix the damaged DNA the same way as an oxygen molecule would do. So this indolzimazole were pretty active as sensitizing the tumor cells. Problem with that, it's fairly toxic. But there are some clinical trials that have been done, and there is some benefit that has been shown with this, especially in male population. If they have high level of hemoglobin and in some type of a cancer variants, there's been some benefit for that. Another molecule in the same family that has been developed was the etimidazole. That was less toxic, but there was no clinical benefit with this one. And then the nimorazole, this one has a neutral group in position 5. It's less active, but much less toxic, and there is some benefit in head and neck. The main problem with the two nitrazole is you can get peripheral neuropathy, and that was not a very pleasant thing to do. And you need to have a very high amount of it to make it effective. So this was a way that to sensitize cells to radiation under hypoxia. Is there now other ways that alternative way to, instead of sensitizing the hypoxic cells, what we can try to do is to selectively kill the hypoxic cells. And this is something that you could do if you have an agent that is going to be activated under hypoxic condition. And that would what we call bio reduction. So one of the best known is mitomycin C, and this is a quinone antibiotic that is can be reduced into hydroxyquinone, and then when it is reduced under hypoxic condition, then it functions as an alkylating agent that would can cross-link DNA and lead to double strand break. But the differential effect between hypoxic and normoxic is rather small. Another compound that has been tried was nitroaromatic compound that could be activated, but there was a lot of normal tissue toxicity, and then no clinical trial was tried with this one. And the one that has most effective in this way is the tirapazamine, which is a benzonatriene oxide, highly selective for hypoxic cells in vitro and in vivo. And I'll show you a couple slides on this one. So this tirapazamine is selectively activated by multiple reductase, mainly the cytochrome P450, can form free radical in hypoxic cells that would induce single and double strand break, base damage, and cell death. So you can see how that would be effective. And it can also sensitize the cells to ionizing radiation by inhibiting the repair of radiation-induced DNA strand break, because it can also inhibit the topoisomerase too. So the way it works is the TPMZ, or tirapazamine, can be reduced in normoxic as well as hypoxic cells, because as long as you have those enzymes like cytochrome P450 that are present, then it would be reduced. What is happening though is that if you don't have oxygen that is present, or if you have oxygen that is present, actually, that the will reverse, and then the oxygen itself then will take this free electron and then reverse the T-parazine to its inactive form. So that's why it's going to be only active if oxygen is not present, leads to the double strand break, and then be effective as a radiosensitizer. It's been used, here's just a cell survival showing the increasing concentration of tirapazamine. It's also known as SR 4233, and this is the difference in survival between hypoxic cells, hypoxic cell here that's much more sensitive to the drug than cells that are growing under normoxia. And here, this is with or without radiation, and same thing here, that you can see that if we have the two together, that the cells, the effect are much more sensitive to killing, and this is more than being additive. That would be the projection here in the dotted line. And then we have the two together here. In vivo, also that was done in mice. And this was done here, where we have, this was the squamous cell carcinoma in mice. And the one that works the best is that the TPMZ is given 30 minutes before radiation. Here, the radiation was used at 2.5 gray twice daily. This is tumor volume with saline alone. This is the TFPXM alone, radiation alone. And here we give X-ray first, followed by TMZ. There's a small advantage of giving it before than giving radiation, but both actually are pretty effective. This was done in 4 different tumor cell lines, not just the squamous cell carcinoma, and pretty much the same result. The problem with that is in the clinical trial is that there's several side effects, a lot of nausea, and the main one is really severe muscle cramping. TPMZ can also work in not only with radiation, but seems to have also a synergistic effect with different anti-cancer drugs. There was a phase 3 clinical trial when it was used with cisplatinum and get a good response rate in non-small cell lung carcinoma. So how do we calculate the effect of a drug? And that's what is called the HCR, the hypoxic cell cytotoxic ratio. So this is the dose of the drug to kill a fraction of cells under normal condition over the dose of drug to kill that fraction when the cells are grown under low level of oxygen or hypoxia. And here are just examples of different drugs. Like I said, mitomycin C is mild. We get about a ratio of about 2. And this is for another region that is similar to that's all a similar electron affinity. This one has HCR 67, and this is this drug here. And then there are terapamazine is a 50-fold. So you have a different range. We have a couple of small formula like this that may come up and show on the board. They ask you what is, give you a question and then ask what is the HCR, and then we'll talk about the dose reduction factor as well as the sensitizing effect of a drug. And those are just small formula that are easy to confuse, but we'll go through that, and it's just keep in mind of what they are. There are another class of non-hypoxic cell radiosensitizers, so those the halogenated pyrimidine. These they may be incorporated into a cellular DNA. The rationale of using a small molecule like that is that they would be that the cancer cells will cycle much faster at a higher rate than normal cells, and they are expected to incorporate these pyrimidine faster than the normal cells. They look like thymidine that you can see here. That then they would expect it to weaken the DNA there because they will prevent the replication of the DNA. And then the problem with this is that you need quite a bit of those pyrimidine to be incorporated in the DNA to have some effect on the cells, because at each replication phase you need to have enough to prevent the replication of a particular cell. So here are just two of them. This is the bromodesoxyuridine that would sensitize cells to ionizing radiation, but can also sensitize cells to UV radiation. And this is, and that's here, this is a clonogenic survival. And this is, you can see that other UV as well as radiation that you could sensitize the cells. The other one is the iododesoxyuridine, and this one would only sensitize the cells to radiation here. Sorry, so this one was radiation and this was with UV. So this iodine is here, and then the BR is on this one here. They need to be delivered in intraarterial infusion because they can be dehalogenated, and they may have some use in some GBM and sarcoma. That would not be recommended for head and neck because they are surrounded by actively proliferating normal tissue. So you would not have much of a benefit of using these as radiosensitizer for that particular cancer. So now I will switch to the second part of this talk, and we'll go over radioprotector. How can we protect normal tissue? And there are different ways that can be done. You can prevent generation of toxic metabolite. We can promote elimination of the toxic metabolite, try to detoxify the free radical, and try to neutralize the aniadog that are forming the metabolite. The one of the first compound I would say that had been discovered that can protect from free radical generated by ionizing radiation was this amino acid, the cysteine, and also the cysteamine. So because they have a sulfhydrid compound here that can be used and to quench the free radical, they can be seen and being a radioprotector. Problem with this is that you need a very large amount to be able to quench all the free radical. It is then toxic, cause nausea and vomiting. But you can get a dose reduction factor of about 1.8. Like I said, they are efficient at being a scavenger of free radical, and then they protect against the oxygen-based free radical. There are other, so these guys, we call them true radioprotector. There are other agents that can lead to an increased blood vessel perfusion indirectly, and then they are not, will give you indirectly some radioprotection. So we talk about sodium cyanide and epinephrine and that sort of thing. But these are not considered like true radioprotector. That would be some sort of an indirect effect due to increased oxygenation, actually, in this case, because we're talking about radioprotector. So here's another small formula. So how do we calculate the dose reduction of a particular factor? So this is the dose in the presence of a drug divided by the dose in the absence of the drug. So this is here you expect to have protection. So you want to have the dose that is going to be less sensitive, then that would be the one that would be with your dividing factor here, right? The best radioprotector that can be at that we have, you can attain maybe a DRF of 2.5 up to 3. This is the best that we can get, and this is to protect the bone marrow in mice. This is how this was determined. And one thing to keep in mind is that even the most effective radioprotector, the efficacy would decrease with increasing lead. So there's a limit to how much protection we can get. So the one that is the most or the best understood and most efficient radioprotector is amifostin. And this was discovered by the Walter Reed following the bomb in Japan, the Second World War. There was a lot of interest in trying to see whether we can protect the population for radiation exposure. And then when the effect of the histamine, the sulfate group was discovered. So they started to look at different compounds and synthesizing more than 4,000 compounds, and then look at what would be the effect of this. So it was found that if the SH group is protected by a phosphate group, so then that would significantly decrease the side effect of this, but still maintain the scavenging of the free radicals. So that's how amifostin was discovered. It is used sometimes in head and neck, and would prevent the effect of dry mouth of radiation. It has to be given 30 minutes before radiation treatment. And then, so that's something that we have to keep in mind, because the generation of free radical then is essentially complete within seconds, right, after radiation. So you have to give it before. It does not cross the blood-brain barrier. It's usually given IV. But there is some toxicity with that as well, nausea, vomiting, malaise. So this drug was the drug that most astronauts would carry with them in case there is a solar flare and they were exposed. The thing that is kind of a little bit funny is that if you have nausea and vomiting and you're in space with a low gravity, that's probably not a good thing to have and see all your thing floating around. But that's the best we have so far. So this is how the amifostin works. It is actually a prodrug that is called the Walter Reed 2721. The advantage of that, it would penetrate tumor at much slower rate than normal tissue. This is what we want. And this is due to poor vasculature, as we talk about, a low pH also in tumor. And another reason why it works better in normal tissue, because alkaline phosphatase is high in normal tissue as compared to capillary. And this is the alkaline phosphatase here. You would have the prodrug that would come here. And then they are inside the cell, so you would have a removal of the phosphate group here, and then release the SH group that we can now go and scavenge all the free radical. It can also, the problem with it is can also scavenge free radical that have been generated by other anti-cancer drug and like the platinum or alkylating agents. So this is a reason why there might be some resistance in using this in combination with radiation therapy, because you want to protect the normal tissue, but not the tumor. Okay, so there are other radio protection mitigation agent. One is the palifermin, that is a human keratinocyte, that is a growth factor, which reduce the incidence of severity of mycosis, and has to be done, can be administered before and after radiation therapy. Potassium iodide will be good to protect if you have radioactive iodine. So that would be something that has been ingested. If you're exposed to a dirty bomb, this would have like no effect. It would be external radiation exposure. Even if you take as many pills of potassium iodine as you want, you'll have like no benefit from this. But if there is some internal injection, so that you could use that to compete out for the radioactive iodine. Another possible protection is to this Salmonella flagellin, which is a bacterial protein that can bind to the toll-like receptor 5 and then activate the NF-kappa B pathway that can increase the proliferation of normal cells from on the gut and hematopoietic cells, and that can then provide some radioprotective effect in those tissue. Some antioxidant can have some potential effect. The problem with that is that you need to take a lot of it, and that has some limited benefit. But there's a study that has been done. If you use a cocktail of antioxidant, you can get a dose reduction factor about 1.2, 1.8 in mice 24 hours after total body irradiation 8 gray. So there is some potential benefit for this. That will have to be used constantly for several days. Okay, so the last part of this lecture, I'm going to try to cover the strategy that we use in chemoradiation and what is the rationale for chemotherapy. And basically, we have a spatial paradigm versus the infield paradigm that we can take advantage of. So for the spatial paradigm, obviously, what we want to try to do with radiation, you need to achieve a local tumor control. And then if you combine that with chemotherapy, then we're going to reach out of field effects. So that would be for systemic disease. And then those are two dependent toxicity. And then you can just get a better control of the cancer progression this way. We can also try to do the infield paradigm. This is where you can try to synergize the effect of radiation and chemotherapy in the tumor, and the effect can be either additive or synergistic or additive, and try to mediate that. We can also now take advantage, and you'll hear from Dr. Ferris later this afternoon, of the radiation to stimulate the immune response, either in combination with other immune checkpoint inhibitor or chemotherapeutic agent, and see how we can benefit from combining those modalities. The rationale is that there are many techniques and concepts that were used in chemotherapy that actually came from radiobiology, the quantitative tumor assay, the concept of cell cycle, and even the sensitivity of cells in different phases of the cell cycle. This is something that were first initiated or presented by radiation biologists, and then also as well as population kinetics. That's something that had been first discussed there. It is more difficult, though, to define what is the dose of chemotherapy than it is for radiation therapy. Radiation therapy, we have a set dose that we know that we want to use and try to get maximum local control, minimize the effect. With chemotherapy, this is more difficult to find the perfect dose. And as well as the variation in sensitivity through the cell cycle, we get a much more dramatic effect with chemotherapy than we have with radiation therapy. And I'll show you why, because we, in a couple of minutes, some of the drugs are going to be like antimetabolite, only effective in S phase, for example. This is a table that I've lifted from the Hall and Gayacci textbook. I'm not going to go to every one of them, but this is just to put in perspective all the chemotherapeutic agents that have been described to have synergy to different level with radiation. And this is their indication, toxicity, and the mechanism of action. So in the next few slides, I'm just going to cover some of them, but not all of this. There is a lecture by Dr. Gail Woloschuk also in one of the links that we've provided that go into more detail of all the drugs. This is a slide I think that I like because it summarizes several of the anti-cancer drugs that we use, where they are active in the cell cycle, and this is like whether they affect the DNA directly or not. So the taxane, for example, they work on the microtubules, which is in mitosis in M phase. And the one that work in S phase, you get all the one that affect the DNA, the synthesis or DNA replication, as well as topoisomerase inhibitor, and so on and so forth. So we have those here. And this slide is similar to kind of sum up the different type of chemotherapeutic agent, what they were working on, and how we use them in benefit. This is also a table that will go through, I'm going to go through some of them in a minute. And this is just, again, to group them into class so you can recognize their mechanism of action and when they are effective in the cells. So the next few slides, I'm just going to go through these guys here. So gemcitabine, it inhibits the ribonucleotide reductase. The androgen receptor can prevent transcription of and DNA repair genes. The taxane, they will bind and microtubule affect their function. HSP90, the HOC protein 90 inhibitor, would inhibit the ribonucleotide reductase, inhibit DNA synthesis, and also the folding of those protein. And then cisplatin or intermediate DNA repair. So just here to show how the gemcitabine works. Gemcitabine, like I said, is an anti-metabolite. It can diffuse into the cells. It goes through several steps of phosphorylation through this kinase using sequential phosphorylation by the desoxycytidine kinase. And so this intermediate here would inhibit, can inhibit the ribonucleotide reductase, which would lead to depletion of the ATP, which is an essential nucleotide for DNA synthesis. So if you have less DNA that is synthesized, you're going to decrease repair, and this is how you increase reduced sensitization as well. One of these intermediate of gemcitabine can also be incorporated in cells into the DNA and then disrupt DNA synthesis as well and lead to cytotoxicity. So there's two pathways, but this is the one that is the most, gemcitabine is most recognized for, would be to inhibit the ribonucleotide reductase and deplete the pool of DATP. The androgen receptor inhibitor, this is an interesting way how we can take advantage of that in combination with radiation. Because the androgen receptor, they are activated by ligand. Here we have like the testosterone, and then it can translocate into the nucleus, and they will activate several genes that are involved in the DNA repair. So if we can use an androgen receptor inhibitor, so that would decrease the pool of repair enzyme that would then facilitate or sensitize the cells to radiation from that point of view. The problem we have if you just inhibit the androgen receptor itself is that not only it would prevent the upregulation of the DNA repair enzyme, but also prevent the regulation of an inhibitor of the AKT pathway. So this is like a yin and yang effect with the androgen receptor inhibitor. So the beauty would be for us when we combine those androgen receptor inhibitor with radiation is that not only the inhibitor are going to prevent the activation of repair enzyme, but reactive oxygen species that is generated by the radiation can activate this inhibitor of the AKT pathway. So then this way will inhibit several pathways all at once by two different mechanisms, by preventing upregulation of those repair enzyme and then preventing the activation of the AKT pathway. So this is the taxane, how they work before. So that would be to inhibit the microtubule formation and then the dissociation of the microtubule through mitosis. And then obviously they will be more effective during the G2M phase of the cell cycle. The HSP inhibitor, the way they work, so usually the HSP help in the folding of protein, and a lot of those protein that needs to be folded are involved in the DNA damage repair or regulator of the DNA damage response. So usually this protein will come and use ATP and be properly folded by the HSP and then be functional. If we use an inhibitor of this or protein that were targeted to folding, then will not be properly folded and then recognized as an unfolded protein to the proteasome and then be degraded. And so if the DNA repair protein are not functional, so then obviously that would increase the effect of radiosensitization in those tumors. The cisplatin, we've talked about that. This is an interstrand crosslink, and following radiation that would then enhance the formation of the intermediate and free radical. They will also prevent the binding of the DNA repair to the damaged strand of the DNA. This is not cell cycle specific, so then it would be effective in every phase of the cell cycle. And as long as there's broken DNA, it will be there and amplify this damage in the sense, and then lead to apoptosis. So a couple of other interesting I think inhibitor that can be used in radiosensitization is the PARP inhibitor. And this one, you probably heard about that with one of the lecture by Dr. Zeman on DNA repair. So the PARP is a polyADP ribose protein. This is involved in single strand break repair. So that would be this pathway here. So if we have a PARP inhibitor, so the single strand break can be then converted into double strand break in the next phase of the cell cycle. But a cell that is normal in the double strand break repair would then be able to repair this. So knowing this, so then we could take advantage of cells that have a defective BRCA1 or pathway, and those are the in the breast cancer, all the BRCA mutated cells. So if we combine the PARP inhibitor for those cell or patient that have an mutation for the BRCA. So when the PARP inhibitor is here, you get a single strand break that will be converted in double strand break, but then there's no functional repair machinery, and then the cells will go to death. So this is the basic principle of synthetic lethality, and this is been described probably in another lecture. And this is the beauty of this is that the cells that have a functional BRCA1 will not be affected by the PARP inhibitor, only the one that have defective the BRCA cells. How do we take advantage of that with ionizing radiation is that if we have a BRCA functional cell, so then we can use ionizing radiation here to help having a second strand break here, and then have cells that could also have increased synthetic lethality here. The BRCA inhibitor also have a different function. They can be increased, in a sense, indirectly increase radiosensitivity because the structure, I don't have it here, but the structure of the PARP inhibitor is very similar to a vasodilator, a drug. So then you can increase the blood flow into the tumor as well. So that would decrease hypoxia and increase sensitivity. There they could generate G2RS following the remodeling of the chromatin that would bring the cells into a more sensitive phase of the cell cycle. So there are different effects of the PARP inhibitor and why it works well with combining radiation and increased sensitivity. This one, I would take that with a grain of salt that has low toxicity. It means that it's not worse than any other chemotherapy drug, but there is still some toxicity with the PARP inhibitor. The ATM inhibitor, this is if you know just one kinase in the field of radiation oncology, this is the one. So this ataxia telangiectasia mutated kinase. This is a kinase that will affect every cycle of the every phase of the cell cycle. This is an upstream kinase that can right away be inactivated with even very low dose of radiation. It can phosphorylate check 1 and check 2 as an effect also on P53. And then these are all the protein that would be affected by this kinase. We can impair cell cycle and impair DNA repair directly or indirectly. So you would think, and actually ATM, so this is how you're going to get cells that are extremely sensitive to radiation if you target ATM or if ATM is defective, because the cells have no way to repair the DNA. Every phase of the cell cycle will be affected and non-functional if ATM is not functional. And this is how the ATM was discovered. This is a disease, ataxia telangiectasia, that mainly affects children that develop secondary tumor. And when they were treated with radiation for their tumor, they were getting second-degree burn, and even some of them died. And so make a long story short, so that was found in 1995 that it was mediated, sensitivity was mediated by a mutation in this kinase here. So now you would think that if we can develop a drug that target this ATM and then take advantage of this, we could increase sensitivity to radiation. There is several drugs that have been, as I say, and currently in development. There is one now, AZD 1390, that is being tried now in radiation therapy in combination for brain tumor. Another one that was not that very successful was the M3541. That was an oral formulation for this kinase inhibitor. They did only 15 patients, 30 grade 10 fraction for escalating dose, but the study was stopped because there was no dose response relationship and no optional pharmacokinetic profile. So this slide here is just to bring into perspective some of the drugs that we have talked about, where they are effective in the cell cycle, which phase of the cell cycles are radiosensitive, and which one are radio resistant. And like I mentioned, some of the drugs here are not cell cycle dependent. So that's good to know. There might be some question on that. So how do we measure the sensitivity of or the enhancement ratio of a radiosensitizing drug? So we just took the dose of radiation of the agent, the biological effect in the absence of the sensitizer over the same dose in the presence of the sensitizer. So this is exactly the opposite of what we have for the dose reduction factor, where we were taking the dose in the presence of the protecting factor over the dose in the absence. So that makes sense because we want to get the here we're going to increase the sensitivity, so that dose is going to be smaller. Let's explain if it works. So the last few slides, I'm just going to talk briefly about targeted therapy. So the best example we have is Gleevec. This is not something that is used in radiation oncology, but it's really like the poster child for targeted therapy, because this is a drug that the disease that is targeted is caused by a single mutation or in a gene, and that's why it's an ideal target. The mutation is caused by a chromosome recombination between the BCR and ABL, and this is the breaking point cluster region that's called BCR. This is on chromosome 22, and ABL is the tyrosine kinase. That is on chromosome 9. So when those two get recombined, you get the formation of an oncogene that constitutively active this kinase protein here that leads to that massive production of the cells here, or expansion of those granulocyte cell. So because we know that this kinase is constitutively active, there was a lot of effort that had been devoted to trying to inhibit this kinase, and to inhibit protein kinase is not a trivial thing to do, because they have a lot of ATP binding pocket that are common. So you want to get selectivity and specificity. So how this was done for the Gleevec is that it was started with a protein kinase C inhibitor, and then through computer modeling, we use its computer-aided drug design, medicinal chemistry, and prediction. You could increase the activity of a particular molecule and then predict where it would bind. And in this case here, it was shown that the active form was like a closed form of the tyrosine kinase, and then this molecule here, which is now the Gleevec, was shown to particularly block the kinase and then prevent autophosphorylation in this way, effectively prevent activation. So with this targeted therapy, we were able to go from a lethal disease, a CML, leukemia, and now we have a more like a chronic disease. So there are several targeted drugs that are been developed and approved by the FDA. Most of them are monoclonal antibody, and those two here are tyrosine kinase inhibitor that target the specific protein or target on tumors. So just the last couple of slides, I just want to go through mechanisms of drug resistance. And so in this slide here, this is shown that resistance can be intrinsic or extrinsic in nature, and then it can be induced at the level of the DNA. For example, if you have a lot of DNA damage, repair protein that will overexpress in the tumor, that can make them resistant to different treatment, including radiation. It could be at the level of RNA. They have some microRNA that are called epimer that can have an effect on epigenetic modification on those DNA damage repair protein, for example. And it could be at the protein level. If you have increased concentration of gene, for example, a tyrosine kinase inhibitor, that would be amplified. There is the most common or most known resistance of anti-cancer drug is mediated by this class of protein here. They call the ABC, the ATP binding cassette that can pump out drug into outside of the cells. And they need ATP, so that ATP can come from the tumor cells, but it also can come from the tumor microenvironment, that they can secrete ATP. That ATP can be internalized and then fuel and activate those ABC transporter as well. The ATP can also work to compete out some small molecule that have been our ATP analog, for example, to prevent activation of tyrosine kinase receptors. So that could be from multiple pathways. This is just, again, for the ATP binding cassette. So there are at least 48 different members of these protein. The multi-drug resistant protein is one of them, and the P-glycoprotein is. So they do have ATP binding site, but they also have binding site for most of the drug that we're trying to treat the cancer cells with, and then they would effectively pump out those drugs. They unfortunately overexpressed in a lot of cancer, kidney, lung, liver, colon, and rectum, which make these cancer cells even more difficult to treat because of that. Okay, so I have about 8 minutes left. So what I want to do here is just give you some example of different question that can come up, and I'm going to give you the answer because I don't have a good way to know if we can do that interactive, but in any case, I can just go through this, and that's something that you can review once you have the answer. So this question is, which of the following treatment concerning hypoxic cell sensitizer and bioreductive drug is true? And I'm just going to go quickly give you the answer here, which is this one that talk about the clinical trial, and that says that it is indicating significant improvement for both local control, overall, and head and neck, and that was true. There is one here, some sort of a trap, and I'm not happy who made this question, because in a sense this is true. Possible clinical trial, this is okay, but here it says that limitation, severe neurological toxicity. So it's not neurological toxicity, it's neuropathy that we have with those drugs. So that's one reason why this was not true. So this one is not true because it's a bioreductive, it's not reductive. Then they're not more toxic in aerobic, that's the opposite. And then so here they're not, so epoxic cells will not be very effective in hyperfractionation because we've shown and discussed before, every fraction of radiation, we're killing more and more of the oxidized cells, and then allow for the reorganization or redistribution of the oxic and epoxic cells. Another type of question is this one: treatment with anti-angiogenic agent may cause a tumor to exhibit increased sensitivity to subsequent radiation dose. And this is due to what? And the same thing here. I'm just going to give you the answer right away, and then we can look through that again. Because like I've mentioned in one of the slides here, so if you use an anti-angiogenic agent, you will transiently normalize the tumor vasculature, and then that will increase oxygenation. And this is probably the best benefit we have for using those drugs here. Okay, and this one here, this is along the same line with SBRT. Toxic cell, the most common cancer below would be here. So then with the same idea here, that you have hypofractionation here, and then an epoxic radiosensitizer will have limited benefit for hypofractionation. Okay. Annotate the share content. Okay. No. Okay. Okay. So, and here we have, amifostem, there's got to be a question. If there's a question about using a radioprotector, it has to be this one. And here's asking, how does it work? So is it scavenging, selecting uptake, reduction of oxygen, stabilizing hemoglobin? And the answer is A and B, because it works as a free radical and then it selectively uptake into normal tissue. Then this is one with they ask for with those little formula that we looked at. So the dose reduction factor, what it would be. So they give you a dose, a percentage survival, and agent X, and then the second dose is required to reduce the survival. So what would be the dose reduction? So the dose reduction would be 2, because you take the one that would be in the presence over the one in the absence. And so this is for that. For sensitization or radiosensitization would be then the opposite, right, the reverse ratio. And the same thing with hypoxia. So what we do here, that you want to have the ratio is going to be using the oxic over the hypoxic cell. Okay, so that's all I have. So let's see if I can stop. Whoops, sharing this. Let's see. Nope. But if I cannot, let's see if I just show. Oops, did I? The speaker. Okay. Okay, I'm trying to stop sharing. Ah, here it is. Stop. Okay. Good. So we have 3 minutes left. And let's see if I have any okay. So I'm not saying any question here, but please keep in mind that all the lectures are recorded, so you can go back and look at all the material, and you can contact us later as well. So if let's see any, so